I'm a pharmacy student, and I've written my diploma thesis with Dr. Haselberger in the field of neuroinflammatory diseases and autophagy, and therefore I'm happy to present you something about this very interesting theme. Okay, what are we going to discuss today? We are going to talk about, as you have seen it, about autophagy in neuroinflammatory diseases, and then I will tell you something about a particular autophagy modulator called the P140 peptide. But first we will start with some terms and definitions. I will talk about the different types of autophagy and the dysfunction in neurodegenerative diseases. It's a little tongue breaker. And yes, in the end, we will talk about the clinical implications, and then we will have time for discussion. And hopefully, if my computer doesn't stop like Dr. Hippe's computer. Okay, yeah. What is autophagy? It's an intracellular pathway that's very important to maintain vital cell functions. And it plays a role in degrading, recycling, and clearing the cell of dysfunctional cellular components, which is very important in neurons, as they don't divide and therefore cannot get rid of these components by simply producing new cells and new neurons. And for example, there are the types of debris that is cleared are damaged organelles, proteins that are abnormally folded or produced in excess, and other types of cell components that the cell doesn't need anymore. Yes. So how is autophagy genetically determined? There is a group of ATG genes that are highly conserved and regulated. So if these genes are hampered, then also the autophagic function is changed, and it may be upregulated or downregulated, both, which is bad for the cell, but we will talk about this later. Yeah, so especially where is autophagy important? I've already talked about it. It's important for the cell so that it can adapt to the environment, that it can maintain homeostasis, so and also for cell development and differentiation, especially for lymphocytes, where you can also see why it is connected with neuroinflammation and the B lymphocytes, which I will also talk about later on. It is activated usually in stress conditions, so if cellular stress occurs, which is in stages of nutrient starvation, hypoxia, or calcium changes, which is in which the cell has to adapt to the new circumstances. Okay, yeah, there are different types of autophagy. There are more than these two types, but I will stick to those two because we don't have unlimited time, as you already wait to present your topics, and therefore I will only talk about macroautophagy and the chaperone-mediated autophagy. Yes, these types I will go on in the next slide, so you can see the differences, but the different components are usually degraded via different pathways, and the size, the solubility, so if a component is lipophile or hydrophile, determine the way. And also, if we talk about the protein misfolding, it's usually the type of protein misfolding, or if it's just a single protein or protein aggregates, that determines how the protein gets degraded. And usually the chaperone-mediated autophagy is used for smaller and soluble proteins. Yes, so you can see on this slide, usually, I don't know, did you see my cursor here? Maybe now, yes. Usually, we have the normal unfolded proteins, and if they are misfolded, the cell tries to repair it. But if this repair mechanism, there are different repair mechanisms, but if they fail, the protein gets marked. And this may be with the ubiquitin ligases, and gets destroyed by proteasomes. This is one pathway to get rid of protein, but it doesn't really have something to do with autophagy here, because autophagy is a different pathway, or in this case, two different pathways. The first one I will talk about is macroautophagy, where there is a membrane forming if autophagy is induced, which then isolates the substances in so-called vesicles, or autophagosomes in this case. And then the autophagosomes bind to a lysosome and form the autolysosome, and then the protein gets degraded. So it's a rather simplistic view, but you get the point. And there is the chaperone-mediated autophagy, which is usually more selective. In this case, the protein gets marked with a marker. It's the key FERQ marker. And these marked proteins are then translocated into normal lysosomes. So there is no formation of the autophagosome. And then they get also degraded. Yes, and these are just two types of autophagy, but you get the point. It's a rather selective process, and is very vital in neuronal function, which is very important in different diseases, which we will talk about. For example, in oncogenetic processes, in metabolic and cardiovascular diseases, and of course for inflammatory processes, which is especially important in neurodegenerative diseases. They are often caused by toxic intercellular protein aggregates and damaged organelles, which then cause inflammatory process via some different pathways. Okay, let's see which diseases are involved there. It's Huntington's disease, especially. I've now specialized on the diseases that are treated in my paper that Dr. Haselberger sent me. So I will go into detail with these diseases. And in this paper, especially NPSLE was treated, which is called lupus usually. And you can see that the autophagic dysfunctions in neurodegenerative diseases both can lead to failure and or hyperactivation of autophagy. In some cases, like lupus, this occurs in parallel, so that in some types of cells, the autophagic pathways are overexpressed, and in some cell types they are underexpressed, and this leads to an unstable condition in which the cells then stay and then lead to inflammatory processes. Yes, this can occur in the central nervous system and the peripheral nervous system, as ALS, you saw it on the other slide, is a disease which is in which the peripheric nervous system is involved. Yes, and the neuronal symptoms that are inclined there is dementia, of course, with Alzheimer, cognitive deficits, as well as sensory and autonomic deficits with multiple sclerosis, and in ALS, also cramps and muscle weakness. All right, so I've talked about the diseases. So you can see in this slide here that in different diseases, the autophagic activity is decreased, and in some diseases it is increased. In ALS, I've also talked about this case here. It is in some cell types increased, and in some cell types decreased. Autophagic influence on these processes is a rather new study field. So, or the study field is not relatively new, but the therapeutic approaches are relatively new. So, as how to influence these processes. But Muller et al. Have synthesized a peptide in which they try to do exactly that, to modulate autophagy. The P140 peptide is a ribonucleoprotein fragment that has a post-translational modification at the serine 140 position, which is generally relatively good accepted and interacts with the heat shock protein 8. And with this interaction, it serves as a partial agonist for T-cell intracellular signaling, and therefore downregulates the autophagic flux, and in turn also the B and T cell activity, which you can see on the next slide, because in the paper, the main focus of autophagy was in lupus. And in lupus patients, which is a chronic neuroinflammatory disease, the patients have hyperactivated CMA and macrophagy pathways. So both types of autophagy are hyperactivated, and especially in B and T lymphocytes, which leads to standing cell activation and neuroinflammatory processes, which then leads to cell death in the further stadiums. Yes, the P140 peptide in lupus significantly slows the peptide loading of the MHC2 molecules, and also reduces the autophagy markers. In the further downstream processes, it significantly reduces the autoreactive B lymphocytes and neuroinflammatory markers. And it does also in mice and in humans, because it's already in a phase 2 clinical trial, significantly betters the clinical and biological manifestations of lupus, which is a very good sign. And they are currently working on the phase 3 trial. Yes, further remarks to P140. It may be also be beneficial in CIDP disease modification, which shows some beneficial effects in a rat model, but these are not published results yet. There are no published results yet. And they have also tried it against the amyotrophic lateral sclerosis model in mice, but it doesn't really show a bettering. And this I think this is really important to know that I don't think, or as I see it, the current state of the current state of the knowledge is not that we can definitely say that autophagy is really the main factor that causes these diseases, but it's rather a part in a pathway, but it's a really important part. But the understanding doesn't really, but it's not completely understood how autophagy works in these diseases. Okay, as I wanted to talk about autophagy in general a little bit also, there are also other autophagy modulators. In this case, they also induce autophagic flux, like rapamycin, nilotinib, and methylthioneum chloride, which you maybe call methylene blau, which is rather interesting that this also interferes with autophagy. And as Dr. Hippe has already told you, there are some natural or food-derived substances with which I've also viewed in my diploma thesis that induce autophagy as a whole, like EGCG, ficitine, or spermidine, and the clinical implications may be that they help in preventing autophagic diseases that are where autophagy is involved. Okay, so the conclusions and take-home messages here. Autophagy, maybe you have never heard of it, now you know it, and it's a very central process in the cellular metabolism. Both up and down regulation can lead to major pathologies. And there are small molecules and other food-derived substances which can activate or inhibit autophagy, and therefore may be valuable as therapeutics or preventive measures in some diseases. And especially in this paper, P140 was treated and it was tested in humans and shows good potential in improving patient outcome in lupus. Okay, yeah, thank you for listening. If you want to read the full paper, you can download it here. Maybe Dr. Haselberger can send you the presentation later on, or the paper. And yeah, I want to open up for discussion a little bit. And the questions I was thinking about, or I was also thinking about while writing my diploma thesis, is how to prevent autophagic decline in these diseases, which are also often connected to a high age, and if it would be beneficial to inhibit or activate autophagy pathways as a whole. As you can see, that this may have also detrimental effects. So yeah, I'm really open to your thoughts, and maybe you can share your comments and thoughts.